Hey guys, today is March 24th, Tuesday. Hope this video finds you all doing well. Um, Today I'm going to start a chapter on genetics. So this is the first chapter that you guys will do um, with this distance learning stuff. I know that it has its challenges. It has challenges for you, just like it has challenges for me. I typically find it a little awkward lecturing to a computer screen, but it is what it is. It's what we've got. And as much as I would love to have you guys here in your desks, that obviously can't happen. So there we go. I'll flip you around so that you can see things a little bit better here. Okay, so I hope you guys are all doing well and staying healthy and staying away from one another. And so we can hopefully get school back in session like we're supposed to. All right, here we go. All right, so chapter 10 or module 10 is just an introduction to genetics. We've talked about genetics before. You know, when you guys were in 7th grade, we talked a fair amount about genetics and we did Punnett squares and that kind of stuff. So I'm hoping that this is a little bit of a rerun for you. We'll go through some stuff. It'll be a little bit more complex, obviously, than it was in 7th grade, but I think you're going to find it to be fairly easily. There's going to be a lot of terminology coming at you. If you need to take a pause, you can pause the video or go back and rewatch stuff if you need to. But it's going to be really important for you to understand the terminology so that it's making sense to you as we're talking about it, because we have to have that language set in order to be able to describe what we're doing. Okay. Also, then we'll talk about different patterns or ways that different genetic things are inherited from our parents. Okay, so 10.1 is about Mendelian genetics, and we call them Mendelian genetics because the whole concept of genetics was really first studied and understood by a guy by the name of Gregor Mendel, and we've talked about Gregor Mendel before. He was a monk, lived in a monastery, and he was the gardener. And so obviously you can imagine back in the 1800s, there's not a whole lot to do. So the gardener would spend a lot of time working in the gardens and trying to understand the types of things that made the plants grow better, longer, or taller, bigger, faster, you know, whatever it happens to be. And he ended up writing all of this stuff down in his, what we would call lab notebooks, but some sort of a journal. And it actually went undiscovered for many, many years until it was like 30 years after he had passed away that someone had discovered all of his notebooks down in the basement of the monastery. And someone actually started going through them and made sense of it. And then with more modern technology, you know, we're talking, you know, 100 years ago, to understand things a little bit better. Okay. So we say Mendel studied pea plants, and he studied a number of different things. And the reason that he studied pea plants or chose them to study among all of the other plants that he could have chosen was that they grew really fast. Okay. They were able to fertilize themselves. They were really easy to pollinate or cross-pollinate, and we'll talk about that in a little bit here. It basically means taking the pollen from one plant and then distributing it to another plant. Obviously, in nature, that happens, right? We have, you know, bees or insects go from plant to plant, and they do cross-pollination. And also that they are true breeding, which means that for the traits that he studies, there's usually only one form of that trait expressed on those plants. Okay. Some of the things that he was studying was the flower colors. So purple versus white, the position of the flowers, axial, which means on the side of the plant, versus terminal, which is up at the top here. If you look at the picture, maybe that'll work better. The seed color, it's either yellow or green. The seed shape, it's only going to be yellow, or I'm sorry, the seed shape is either round or wrinkled. Sometimes this is inflated or constricted when we talk about pod shape. the pod color. The pod could either be green or yellow. And then the length of the stem, it could either be a tall or short or what we call dwarf. So we give him the credit for being the father of genetics because he's the first, the person that basically came up with this stuff. Granted, a lot of people have helped the understanding of genetics. Sorry, I just wanted to check to make sure real quick that I'm recording because the other day I recorded a lecture for an hour and then realized I had never hit the record button. So it was a wasted hour. Okay. So genetics then is the study or the science of how genes are passed on from one generation to another. Some plants are only producing, and part of the reason he liked pea plants is because they only produce green and yellow, right? They're not producing some weird in-between green, like in between a green and yellow color. It's either going to be green or it's going to be yellow. It's going to be tall or it's going to be dwarf. It's going to be inflated or constricted. It's either going to be wrinkled or round. Those traits that he's studying were specific, and it was really easy for him to sort of trace how these things or these traits, the characteristics, get passed on from one generation to the next. He cross-pollinated plants depending on what he was looking to get, and he would take the gametes from one plant and fertilize them with the gametes from another plant, and he would look at the outcome and see what happens. So when he did that, he took one green and one yellow plant, bred them together. Those are what we call the parental generation. You can see over here in the picture, there's a yellow and a green. Those are both pure breeding, okay? And in the first filial generation, or the F1 generation, all of the offspring ended up being yellow. Now, if we have a yellow and a green, doesn't it seem like you would at least get one green or maybe a part of a little bit of greenish color? But that wasn't the case. And so this is sort of where he started to become perplexed. Like, okay, if I mix yellow and green, how come I only get yellow? So what he did then was he let those two self-pollinate, and he bred them. So they bred themselves. And then in the F2 generation, he ended up with a ratio of 3-to-1. All of a sudden, a green pea showed up. Okay. So now he's going, well, why only one? And every time he did it, he ended up with only one green. Okay. So now he's getting really curious, and it turns out that that's just the ratio that it works with, or that it goes through. Okay. The parental generation, a green and a yellow, all of the offspring are yellow. In the F2 generation, we always get this ratio of 3-to-1. Okay. And we'll understand why in just a little bit here. So the fact that he's getting these two different forms, green versus yellow, he starts to realize that there's got to be some alternative form of that gene that's getting passed on from generation 1, or the F1, to the F2 generation. Okay, so the gene, those little alternative forms are what we call alleles, and we'll talk a lot about alleles. Okay, that's one of those terms that you need to get into your head because we're going to talk a lot about it. Okay. So the gene for the yellow seed is different than the gene for the green seed. And because of the ratio of 3-to-1 that kept coming over and over and over, he determined that something must be dominant there. There must be something that's dominating over the green. Okay. Now, this might sound familiar to you from 7th grade. Dominant versus recessive alleles. Okay. One of the alleles masks the result from the other one. Okay. So the trait that appears in the F1 generation, okay, the F1 generation over here, they're all yellow. So yellow must be dominant to green, okay? It is masking the result of the other allele, okay? The dominant allele masks what happens, or what the recessive allele would have, or the form. The trait, we'll talk a lot about traits. Okay, the recessive form is the one that's being masked, okay? So in this situation, we have the yellow is dominant and green is recessive, okay? Now, he knew that two alleles were responsible for the color of the peas, okay? Yellow seed masked the green, so he labeled it dominant, okay? And you'll notice that it's with an uppercase Y. Green seed was also labeled Y, but it was a lowercase y, all right? So we'll probably refer to these as big Y and little y moving forward. So this brings him to the point where he's determining whether the organism is either homozygous or heterozygous. Okay, now remember, homo means the same. Okay, so if they're homozygous for yellow or dominant, okay, so homozygous dominant, that means it's big Y, or uppercase Y, uppercase Y. Okay, so some are homozygous dominant. If it was homozygous recessive, it would be little y, little y. And then there's also a heterozygous condition where you have an uppercase and a lowercase y, so big Y, little y. Okay, hetero means different, homo means the same. Okay, homozygous, big Y, they're the same. Little y, they're the same. That'd be homozygous dominant versus homozygous recessive. Okay, so he also came, we also have these ideas of what's called a genotype, and we say genotype. I want you guys to be thinking about the gene, okay? That's the letter that represents the allele, okay? So in the case of seed color, we're talking about big Y, big Y, little y, or little y, okay? These are the genotypes for seed color. The phenotype then is what we actually see, the things that we observe. Maybe it's I mean, yes, we're talking about color, so obviously we can't feel the difference between yellow and green, but it's it's a visual cue. So is it yellow or is it green? Okay, each one of these, flower color, position, seed color, shape, pod color, stem length, each one of these things is a different phenotype caused by a particular genotype, if that makes sense. Okay, I mean, I hope it does make sense, because this is what we're talking about, and we'll be referring to phenotypes and genotypes an awful lot. Okay, so he used homozygous yellow and green seed plants in his parental cross. Okay, so yellow and green, one was homozygous dominant, one was homozygous recessive. During meiosis, it splits the chromosomes. It leaves one chromosome in each gamete. Okay, so recall those gametes are haploid. They have half of what they will have once they're fertilized. Okay, during fertilization, or I'm sorry, the chromosome in each one could either have the big Y or the little y, right? So depending on which one each had, as they fertilized, okay, as they became fertilized, one was a big, one was a little, and now those are united or reunited to make an offspring, okay? The law of segregation then, when we think back to meiosis, tells us that two alleles for each trait, okay, they will separate during meiosis, okay? Now, depending on how they line up on the metaphase plate, okay, they're lined up here, they'll get pulled apart, and this is, the law of segregation is relatively random, and it's going to separate those two chromatids that are going to code or have the information for the same traits, right? One is going to go, so maybe it's a big Y, little y, so one Y goes this way, the other Y goes this way, each one of them creates their own new cell, and then they go through that process again to make the gametes. Now, a hybrid is characterized by this condition where the organism is heterozygous, okay? If they're heterozygous, that means that they have both the dominant and the recessive allele, okay? Now, oftentimes you think about a hybrid, it's like, oh, I got my dog is a hybrid. It's a mix between a lab and a poodle, and it doesn't shed, or, whatever it happens to be. Okay, now this is a liger, right? It's a mix between a lion and a tiger, and it has traits of both. So we also kind of call this a hybrid as well. But when we're talking about genetic hybrid, we're talking about having both the dominant and the recessive alleles. So again, another way of a natural way of things sort of mixing and giving genetic diversity happens during meiosis, and that's the law of independent assortment. When the chromosomes line up along the metaphase plate, they line up completely randomly, okay? And because they line up completely randomly, it evenly distributes the chromosomes and the traits that are on those chromosomes, or the genes that are on those chromosomes, right? So those homologous chromosomes, they separate during meiosis 1, and then depending on how they lined up on the equatorial plate or the metaphase plate determines how they're going to be separated, okay? One gamete is going to have the green allele, the other gamete, the yellow allele, okay? So in order to predict the different traits that offspring might potentially have from the parental generation, we have what's called a monohybrid cross, okay? So we're crossing just one set of genes, or the genotype from parent 1 against the genotype from parent 2. We'll probably refer to it as mom and dad. I mean, that seems to be the most easy, understandable way. Or the mother and the father, right? And when we set up Punnett squares, we don't really care if we put mother on top, father on the side. It really makes zero difference at all, okay? So the monohybrid cross then is what we're doing, and this is a hybrid for one single trait, okay? We can do what's called a dihybrid cross, and we'll talk about that in a little bit. It's a bigger Punnett square, and it, you know, instead of 4 boxes, it has 16, and things get a little bit more complicated. It's not impossible to do. And then they have a trihybrid cross, which is really complicated to do. I did one in college for bonus points. Okay, so the possible genotypes now, if you remember back to when we did Punnett squares, you drop one down from the top and the one from the side. So we get a big T and a big T, right? So the genotype here is big T, big T. Here we have a big T and a little t, and we always write the capital letter 1 first, okay? The dominant allele first, okay? So you have big T, little t, another big T, little t, right? Because we're dropping these two into the same box. And then finally we have a little t and a little t, okay? So the possible genotypes from this monohybrid cross are big T, little t, little t, little t. Let me back up. I think I said that wrong. Big T, little t, big T, little t, little t, little t, okay? Again, big T, little t, big T, little t, little t, little t. I know this sounds kind of confusing to listen to, and sometimes, you know, I have said this so many times over the past that it just kind of is in my head, and I say it fast, but just pay attention to what I'm saying and try and understand where I'm getting that from. Okay, so the genotype ratio then, there are 3 different genotypes, right? There's big T, there's big T, little t, and there are 2 of them. And then there's little t, okay? Those are the genotype, the possible genotypes, okay? So the genotype ratio then is 1 to 2 to 1. All right, are you with me? That's the actual genes. The phenotype, okay, the part that we can actually see and observe, that's the phenotype. There's only 2 phenotypes, right? You're either tall or you're short, or tall or dwarf, I guess I should say. Yellow or green, okay? The phenotype ratio on this monohybrid cross would be 3 to 1. You're gonna have 3 yellows and 1 green. You can come in. I'll just pause for a second. All right, here, let me get back at it here. Mr. Henriksen just came in for a little bit here. Where were we at? Okay, so we were talking about the monohybrid cross. Again, these are heterozygous, uppercase and a lowercase, or a dominant and a recessive. When those monohybrid crosses breed, these are the possible outcomes, okay? Tall, short, or dwarf, okay? So the genotype ratio is 1 to 2 to 1, 1 to 2 to 1, and the phenotype ratio is 3 to 1 because 3 are tall and 1 is short. Now, it's probably worth mentioning that it doesn't really matter which letter you use. We could use the letter Z to represent tall versus dwarf, or blue versus green, or whatever it happens to be, but sometimes it's just easier to use the letter of the dominant trait. So this brings us then to a dihybrid cross. This is when we cross to test for 2 or more traits. So in this one, we're looking at wrinkled versus smooth and yellow versus green. Wrinkled is dominant to smooth, so we would expect there to be, and then yellow dominant to green. Okay, so the results of this cross, and I'm not gonna go through each of those, but you can see all of the different potential crosses there are. There's 16 different combinations that there could be, okay? But of the phenotypes, there could be 4 different phenotypes: yellow and round, yellow and wrinkled, green and round, or green and wrinkled, okay? But there could potentially be 1, 2, 3, 4, 5, 6, 7, 8, 9 different genotypes, okay? So the ratio for the genotypes then would be 9 to 3 to 3 to 1. If we were to look at these, that's what it will look like. But there's only 4 phenotypes, okay? Yellow and round, yellow and wrinkled, green and round, green and wrinkled. Okay, so the Punnett square was developed in the 1900s. Gregor Mendel didn't even know about Punnett squares. He was doing all this work, and then people came up with this stuff later. We've talked about Punnett squares before. Basically, what we do is we take the alleles, we put them on, we make a box, and then we put the alleles on there, and then we cross them together. And then we can look at the possible genotypes, and then from the genotypes, we figure out the phenotypes, right? Based on what's dominant versus recessive. This is what a dihybrid cross would look like, or the Punnett square would look like if it was a dihybrid cross. Now, when we think about the probability of things happening, it's like flipping a coin, right? If you guys were here, everybody would have a coin, and I would make you flip it 100 times, okay? What is it, heads or tails? Tell you what, if it's heads, you can stay home tomorrow. If it's tails, you can stay home tomorrow. See, you guys win both ways. It's heads, so you guys get to stay home tomorrow. Okay, so if I flip the coin, what are the chances it's going to be tails? 50-50. Okay, now when I flip it again, what are the chances of it tails? What are the chances? Yes, you're right, 50-50. And as it turns out, it is tails. Okay, every single time you flip a coin, you have a 50-50 chance. Okay, if I were, now to make this make it a little bit different, if I flipped it and I got tails, there was a 50-50 chance of that happening. If I flip it again, there's a 50-50 chance of getting tails again, but getting tails 2 times in a row, there's a 25% chance. And then flip it again, and there's a 12.5% chance of getting tails one more time. And then flip it again, and then flip it again. Every time, this is a statistics thing, the odds of getting that many in a row go down and down and down. Like the odds of getting 6 in a row tails is really, really low, like a fraction of a percent, okay? Does that mean it never happens? Well, of course it can happen. It's just that the probability of that happening is relatively low, okay? Like some people have people that have kids. Maybe you know a family that has 4 girls or 5 boys, and they have 5 boys in a row. The odds of having 5 boys in a row is really, really low. But every time you have a kid, it's either got to be a boy or a girl, right? So there's a 50-50 chance, but the odds of having a boy and then another boy and then another boy and another boy, so forth and so on, every time it goes down and down. Okay, so that was the basics of the Punnett square. I'm going to pause for just a second here. Okay. Share screen again. Sorry about that. Okay. So using the Punnett square is how we make predictions of how those organisms are going to let me do this, because I didn't really explain how to do the Punnett square very well. So let me just do this real quick. We will, a lot of times, or in your worksheet, there will be questions about like, if you have a, let's just talk about eye color, brown versus green eyes. Okay, so brown will be represented by a big B. So if we have a homozygous dominant mother and a homozygous dominant father, we cross those, it's pretty easy to see what's going to happen, right? Big B, 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 big B. Every single one of the offspring is going to have brown eyes then, right? None of them will have green. But if we do this, now the mother is heterozygous, the father is heterozygous, but they both have brown eyes. So then we end up with this. We have big B, big B, big B, little b, big B, little b, little b, little b. Okay? This offspring is going to have brown eyes. This one is going to have brown eyes. This one is going to have brown eyes. Because all three of these have the dominant allele, and if you have the dominant allele, it masks the stuff that's happening with the, or it masks the effect of the recessive. This one is going to have green eyes, okay? Because there's no dominant allele taking over, so this one will have green eyes, okay? So what are the odds or the probability that the offspring, let's just say two people have a baby, what are the odds that that baby is going to have brown eyes? 75% chance, because brown is dominant to green, okay? So if the offspring has blue eyes, or I'm sorry, green eyes, there's a 25% chance of that happening, okay? So we can make predictions about what's going to happen based on what we find in these Punnett squares or looking at the probabilities. Okay, so that was it for 10.1. I'm going to keep going here along 10.2. 10.2 is about genetic recombination and then gene linkage. Genetic recombination is happening when we have genes that are produced from crossing over an independent assortment. I mean, they're not created from that, but it changes the combination based on how the crossing over an independent assortment works, or how it occurs. Now, you remember crossing over, the two chromosomes cross over, and then a segment from one cuts off and attaches to a different segment. So there's that. And then how they line up on the metaphase plate and then subsequently are pulled apart. That's what causes independent assortment. Okay, those, again, are affected by how those homologous chromosomes are lining up, and then the sections and how those sections of DNA are exchanged during crossing over, section of the chromosome, I guess I should say, okay? So gene linkage occurs when the genes are close to each other on the chromosome. It's less likely that they're going to be split up during crossing over because they're physically closer to each other, okay? And the closer they are physically, the more likely those genes are going to stay together. And it might be the genes for eye color and the genes for hair color, okay? And if those two are close together on the chromosome, then they are going to exhibit what we call gene linkage, okay? Now, a chromosome map then happens when we can actually look at the chromosome and we can see where crossing over is occurring. And oftentimes it happens more frequently between genes that are further apart, that they get separated, right? Because if crossing over happens from this section and beyond, then it's more likely for those things to not be, not to have gene linkage because they are so far apart. So other conditions that happen with the chromosomes are like polyploidy. That means poly means many, mono means one, di means two, tri means three. So this polyploid that we see over here is actually what we call triploid or triploid. This one is diploid. Or, I'm sorry, yeah. because there are two of each one. Haploid, again, that's when there's only one set. Normal cells are diploid, right? Some plants are polyploid, and plants that are polyploid can have traits like they have increased vigor, meaning that they're stronger, they grow faster, they're healthier, they have more balanced growth, things like that. Okay, that was a really short section, obviously. Now we're on to 10.3, which is applied genetics. All right, selective breeding. I think all of us are probably familiar with selective breeding. And the reason I put the dog thing up here, or the dog image, is because that's probably one area where we really see it a lot in our daily lives. Now, this happens obviously with plants and all sorts of stuff, but a lot of people tend to keep dogs around, and so it kind of makes sense. So selective breeding happens when we have traits of an organism that we like, and for whatever reason, we like those traits, and then we select the organism for those traits, right? So if we want a dog that is, friendly and maybe doesn't shed, then maybe we want to pick like a golden retriever or a Labrador retriever, and we crossbreed it with, say, like a poodle, because poodles have hair, they don't have fur, so they don't shed. Okay, that's what selective breeding is. We take the traits that we like or that we find desirable, and then we artificially breed those organisms together in order to get the desired traits that we're looking for. Now, this has been happening, for centuries and thousands of years, and it started with wolves, right? So probably what happened is, as man started using technology, or I shouldn't say, humans started using technology, or human-like species started using technology to help them hunt, like spears and things like that. They were able to spend less time hunting and foraging for food, and more time socializing, like hanging around a campfire, things like that. And as the story goes, the wolves are curious organisms, and the wolves came, and maybe one of the wolves had pups, and one of the humans kept one of the pups, and then that pup became impregnated. And so they basically, what they did is take all these pups and then keep them around, and then they become domesticated, and they don't necessarily take off, and you could use them for pulling dog sleds and things like that. And then eventually, thousands of years later, we have a situation where we have dogs that are teeny tiny, and we have dogs that are really big, and we have some dogs that are good at hunting, and some dogs that are really good at swimming, and some good dogs that are really good at being guard dogs, and some dogs that are really good at pulling sleds. Okay, and that's a product of selective breeding. Hybridization happens then when you cross two parent organisms that have different traits. And when I say different, I don't mean like one is green and one is yellow. I mean like completely different characteristics, like a tomato plant, and you have one variety of tomato plant that grows really fast, and one variety of tomato plant that is resistant to some sort of a disease. Okay. Well, if you are able to breed those two together or do selective breeding, you can hybridize that organism, and now you have a tomato plant that is a hybrid that grows fast and it's resistant to disease. Okay, the infographic that we're looking at the right over here. The infographic on the right here, it's showing plant that is resistant to disease A and B. And so what's happening then is we're breeding this plant over here that is resistant to disease C, and the offspring then in the F1 generation, that hybrid is now resistant to A, B, and C. Another thing that we look at is what's called inbreeding. This is what happens when closely related organisms are bred together for desired traits. Sometimes it happens simply because of proximity. Like a wolf pack. Inbreeding is a very, very common thing in a wolf pack because, the mother has babies, and then those babies are also part of the wolf pack, and they don't necessarily recognize each other as like a brother and sister or cousins or second cousins or whatever it happens to be, but they're closely related. Now, when inbreeding occurs artificially because humans have traits that we're desiring in an animal. Like, for example, the Clydesdale initially came from Scotland, and it was inbred because of number one, large size. It's very muscular, powerful. It's agile and very obedient. So it was bred as like a workhorse, obviously workhorse for people that worked on farms and things like that. Well, it also, obviously, inbreeding has its disadvantages as well. Sometimes harmful traits reside in those genes, and if inbreeding occurs, then those harmful traits get passed from one generation to the next, and there aren't necessarily new genes to come in and take over for those. One place that we really see that happening is in the royal family of England, or Great Britain rather. We'll talk about that in just a second here. Okay, so that was it for 10.3. 10.4 is about basic patterns and how humans inherit different traits. So if you're going to continue on, go ahead. If you're not, just go ahead and pause your video and pick it up another time. All right, so there were two scientists, Garrett Archibald, well, Mendel, let me back up a second here. Gregor Mendel's work was, again, put away. It was written down in little notebooks, and it was put in storage underneath or in the basement of the monastery, and it was forgotten about for 30 years. Well, it wasn't until a scientist by the name of Archibald Garrett started having patients. He was a doctor, a physician, and he started having patients that had black urine. And it turns out that they have a condition called alkaptonuria, and it is occurring because of an enzyme deficiency that causes the urine to be black in color. Okay, it started out when the individual was born and continued through their entire life. Well, it was determined later that this is a recessive genetic disorder, a genetic disorder, not just some environmental something that was happening, but rather a problem with the genes of those human beings. So they started digging into this work that Gregor Mendel had been doing years ago, and then they started coming up with, and they started thinking, geez, Gregor Mendel was really onto something here. And it was through Gregor Mendel's work that they were able to come up with a lot of advances in the field of genetics. Okay, so an individual then who is heterozygous, okay, so they have both the dominant and the recessive allele, is what we call a carrier, okay? So if you're heterozygous, you are a carrier because you have the recessive allele. So you have the little a. Now, the individual, unless it's the dominant allele will take over or mask the results, or what would happen if you only had the recessive allele, okay? One dominant one will take over for the trait. Some other things that alkaptonuria was causing were these black spots in the sclera of the eye, and then also on the hands, certain areas in the hands are darker colored. Okay, so we can look at what's called a pedigree, and this is a diagram tracing how these traits are inherited through several generations, okay? Now, squares represent males, circles represent females. The horizontal line then are all of the horizontal line between the two are the mates, okay? So this would be father and this is mother. They're connected by a horizontal line. And then we have a vertical line that's connected to a horizontal line. Now, anyone that's connected directly to the horizontal line with a vertical line is the child of these parents, okay? So this is the parental generation. This is F1 generation. This is F2 generation, okay? So this is a son of the original parents. This is a daughter of the original parents. This is a son of the original parents, daughter of the original parents, okay? Now, the person that's connected on the horizontal line between those two then is their child's wife or husband, okay? Does that make sense? The vertical lines connect to the offspring. The shaded ones are the ones that have whatever trait or are affected by whatever particular trait we're talking about, okay? I mean, it could be something like brown eyes. It doesn't always necessarily have to be something bad, okay? So Here's the mother, here's an affected son, or I'm sorry, affected daughter, and another affected daughter, but the sons don't have whatever it is. Notice then, when the son and his partner have offspring, none of the offspring have that condition, okay? Well, when we look at the mother that is affected and the father unaffected, 2 of the 4 kids are affected, okay? And again, over here, 1 of the 2 kids is affected. So really, that's just more to help you to understand how to read the pedigree, okay? The phenotypes and genotypes can be predicted using pedigrees, and we'll talk about 3 different ones. Tay-Sachs pedigree. We're looking at a Tay-Sachs pedigree, and we'll talk about Tay-Sachs in just a little bit here. It's a recessive genetic disorder, so it tends to follow around the recessive gene, obviously, okay? Carriers will not express the trait. You would have to be recessive for both of them. Okay, so a recessive genetic disorder happens when individuals have the recessive allele. So they have to have both of the alleles are recessive, okay? Both lowercase. The parents of the offspring both are carriers in this pedigree, right? So mother is not affected by the trait. Father is not affected by the trait, but they're both carriers, meaning that they're heterozygous. Okay, they have 1 big Y and 1 little y, if that's what it happens to be. Okay, well, they have offspring. The daughter is a carrier but is unaffected. The son is a carrier but unaffected. The other daughter is not even a carrier. She's unaffected. And then there's a son that is affected, right? So if that son is affected, they're obviously not considered a carrier. They're considered to have it. So they have the recessive form for both of the alleles. I mean, there are 2 alleles, and both of those 2 are the recessive form. All right, the first recessive genetic disorder we're talking about is cystic fibrosis. It's a somewhat common genetic disorder, especially among Caucasian people or individuals. It affects how mucus is produced in the lungs, and digestive enzymes, and then the sweat glands. Sometimes people with, or oftentimes people with cystic fibrosis, they end up passing away earlier on in life because they end up having chronic pneumonia and respiratory issues. You know, so they have relatively limited lifespans. When I say limited, I mean, they could live, 20, 30, 40 years old, but much beyond 40 or even 50 years old is getting to be less common because of all of the chronic respiratory issues. Albinism, that happens when there's an absence of pigment, or what we call melanin, in the skin. Those individuals have white hair, pale skin, and pupils, and they obviously are very sensitive to the sun and typically suffer from vision problems. Tay-Sachs is the next one. Tay-Sachs happens because of abnormality on chromosome number 15. Okay, they end up with what's called a cherry spot in the back of their eye, and this is predominant with individuals of Jewish descent or Eastern European and Jewish descent. Absence of caused by absence of an enzyme used to break down gangliosides, which is the accumulation of lipids, which are fats, and it causes some mental deterioration. Okay, so that's it for the recessive genetic disorders. The dominant genetic disorders are obviously caused if you have the dominant gene. Now, people might think that if it's a dominant genetic disorder, then a lot more people should have it in the population of humans. But realistically, if it's something that's bad, process of evolution is going to weed that out of a population. We'll get into that more when we talk about evolution and how these genes affect that. But most individuals then would be homozygous recessive for those. The first one we'll talk about is Huntington's disease. That affects the nervous system. There's about 1 in every 10,000 people in America suffers from Huntington's. Those symptoms usually start to to show themselves between the ages of 30 to 50 years old, and it causes gradual loss of brain function, uncontrollable movements, difficult for them to control their emotions. There is no cure, but you could be tested for it to see. You would have to provide a DNA sample, and they'd be able to determine if you could potentially get Huntington's disease, which then leads you down a different rabbit hole where you're thinking, should I get tested to see if I'm going to have these diseases? I personally would say no, because I wouldn't want to know if my body and mental health was going to deteriorate once I turned 40 years old, which happened a couple of years ago. So I just don't think I would want to know. The next one is what we call achondroplasia. It's a form of dwarfism. It results in smaller body size and limbs. It affects 1 in every 25,000. Those individuals have a normal life expectancy, and 75% of people that have achondroplasia have parents that are average size. All right. That was 10.4. Last section, 10.5, and we're talking about complex patterns of inheritance, or how traits are passed from one generation to the other. Okay, so there's a condition called incomplete dominance, and this happens when the heterozygous phenotype, so here we're looking at little r, big R, big R, the heterozygous phenotype. Now, if these two breed together, you're only going to get one allele. And if it's the white flower, you're getting a little r. If the one from the red plant is a big R, when those two come together, you end up with a heterozygous phenotype because you had homozygous parents. Homozygous recessive, homozygous dominant, okay? In the F1 generation, every single offspring then is heterozygous. And in the heterozygous condition, under incomplete dominance, there is an intermediate form of that trait, okay? So a red snapdragon and a white snapdragon, when they are bred together, result in a pink snapdragon, okay? So that intermediate form. Codominance then is a condition where both of the alleles are expressed if they're heterozygous. So these are both homozygous, okay? I don't know which one is dominant over the other. One is homozygous recessive, one is homozygous dominant. But the offspring, if these two have offspring, the offspring then have to be heterozygous. And that heterozygous offspring leads to traits again from both. So both of those traits are being expressed. It's not like if this was incomplete dominance, it would be the entire fish would be some shade somewhere between a blue and a red, but it's not. It's codominant, so we see blue with some red as well. Sickle cell disease is an interesting one. We've talked about sickle cell disease before, but sickle cell, if you have sickle cell in you're heterozygous, that means that you have some normal blood cells and then you have some sickled cells. Okay, they call them sickle because it looks like a sickle, like would have been used in the fields centuries ago. Some interesting things about sickle cell is that the prevalence of individuals that have sickle cell trait reside in areas where malaria is common, and individuals that have sickle cell trait are immune to malaria. People that don't live in those warmer regions are more likely to not have sickle cell trait. So that's very clearly a sort of an evolutionary thing to protect individuals from malaria. Okay, the next one is what we call multiple alleles. That happens when you have more than two alleles that control a particular trait. You know, we look at rabbits, and they can have different colors of hair depending on what their genotype is. So it could be a range anywhere from white to white with black feet and ears to grayish to what we call wild type. Wild type would be like a heterozygous, except that it usually has the brownish color. Blood types are controlled by three different alleles or multiple alleles. Okay, there are three alleles that control blood type: A, allele B, and allele O. Okay, but the possible blood types that you could have are A, B, AB, and O. So there's four possible blood types, but there are only three alleles that control blood type. Interestingly, we talk about donating blood and things like that. And, if you have blood type O, that means that you are the universal blood donor. You can donate blood to anybody. If you are blood type AB, you can receive blood only from people with type O. But people with type O, I'm sorry, if you have AB, you can receive blood from anybody. But if you have type O blood, you can only receive type O. If you get a blood transfusion and any of these proteins, the type B protein, the type A protein, gets into your body recognizes it as a foreign invader, and it starts attacking your body's blood cells, and can result in death, as a matter of fact. But if you have type AB blood, your body already recognizes the type A and B antigens, or those proteins that are on the outside of the blood cell. So you can get blood from anybody, it doesn't matter, or from any of the three blood types. So this is just a look at the population. 48% of Americans are type O, 37% are type A, and then, a small percentage are AB, and a small percentage are type just B. Epistasis is the next condition we're going to talk about. It happens when one allele is hiding the effects of other alleles, and this is the what brings about the different colors of Labrador retrievers, right? Some are black, some are brown, some are golden colored, and some are more white colored. And then there's sex determination genes. You've probably heard of X and Y chromosomes. The sex chromosomes are the ones that determine the individual's gender. Okay, so you have two sex chromosomes. Well, you either have an XX or an XY. Okay, females are XX, males are XY. Probably the reason females have two X's is because there's that little extra area on the chromosome where all of the information needed for making reproductive organs are probably located on those those sections of genes. There's also autosomes. Those are all of the other chromosomes. Okay, there's 22 pairs of chromosomes. Okay, and two sex chromosomes. Dosage compensation happens when one X chromosome in females is inactivated. New research is suggesting that some of the inactivated genes are more active than what were previously thought. Okay, so here's the sex chromosomes XX versus XY. And then there are also some traits that are what we call sex-linked traits. These are controlled by genes that are located on the X and Y chromosomes. And because of that, I mean, if those are the sex determination chromosomes, it makes sense that those are the ones that are going to be responsible for those traits. Like males are more likely to suffer from sex-linked recessive disorders because they're being masked by the effect of the other X chromosome. So basically what I'm trying to say is that if there's a negative trait coming off of this, if you're female and there's some negative trait that's coming off of the one chromosome, there's possibly a chromosome on the other side that can overtake for it, so that it doesn't have that harmful or negative trait. Some of the things that are sex-linked would be color blindness. It's a recessive X-linked trait. 8% of males are affected by color blindness, and very few, like it's pretty rare in females to be colorblind. Hemophilia is another one. And I spoke earlier about hemophilia in the royal family. You'll notice that there is a high or a significant amount of people within the bloodlines of the royal family that have hemophilia, and that's a blood clotting disorder. Now, Individuals that are in the royal family tend to want to keep royalty in the family. So there tends to be a fair amount of inbreeding. And I'm not saying that brothers and sisters are getting married and having offspring, but certainly a version of cousins, back in the day. It's not terribly socially accepted nowadays, for cousins or second cousins to get married, or whatever the relations happen to be. But part of the reason that um, versions of inbreeding isn't good is because there can be negative traits, and those negative traits then end up being passed on from generation to generation, and there aren't new fresh, or I shouldn't say fresh, but different genes being introduced into the population that intermix and then thin out the occurrence of genes for hemophilia. Some traits are polygenic. Poly means many, so this means that they are controlled by many genes. Things like, or multiple pairs of genes. Things like skin color, height, eye color. Those things are all controlled by a whole bunch of different genes. We talk about brown eyes versus green. Realistically, it's not that simple. Those traits are controlled by many genes. Then there's also environmental influences. Sometimes the environment affects whether or not, some sort of the organism is going to have some sort of an effect. Scientists were doing tests on rabbits. What they did was they took a rabbit, they shaved the hair off the rabbit's back, and then they put an ice pack on it. And then the area where the ice pack was, because it was cold, that area started to grow darkly colored hair. Now, if you think about that, why would that be important? Areas that have higher blood flow, areas that are more susceptible to being cold are going to turn darker in color because they'll get more thermal energy from the sun, right? Just like wearing a black shirt on a hot day versus a white shirt, okay? If it's wintertime and you're an organism that lives outside, you probably want to get as much heat from the sun as you possibly can. And so that's one example of an environmental impact. Depending on the amount of sunlight or the temperature and the environment, some plants won't flower. Sometimes the leaves fall off prematurely. And those are different types of environmental impacts. One really great way of studying the effect of genetics on the environment is by studying identical twins, because identical twins literally have exactly the same genes. So what happens is that after the sperm and egg are fertilized, then that egg splits, and each one of those eggs, then usually they stick together, but if they don't stick together, each one of them implants in a different part of the uterus, and then you have identical twins. Fraternal twins are different. Fraternal twins happen when the mother releases 2 eggs instead of just 1. Okay, so then the genetics aren't identical. But after fertilization occurs, once that egg splits, those genes, or the DNA in that egg, is exactly the same as the daughter egg that comes next. Okay, so that's it. Listen, if you have any questions, please get a hold of me. I'll be available during the times that you'll see. You'll have a schedule. We'll obviously be able to see your schedule, and I'll be available during those times, or even before or after, or whatever. If you guys, I'm pretty good about checking my email even in the evenings. So if you're working on stuff in the evening and you have questions, please shoot me an email, and I'll try and get back to you as soon as I can. Other than that, I hope you guys are doing okay. I know this is a tough time. It's, you're probably getting bored. Just hang in there. We'll be through this soon enough, and life will go back to normal. All right. We'll talk to you guys soon.